This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 71 for February 26th, 2010. Hey everybody, it's Vincent Racaniello, and this is TWiV, This Week in Virology. And today's motto is neither snow nor rain nor heat nor gloom of night stays TWiV from the swift completion of its appointed rounds.
I couldn't have said it better, Vince. I could not have said it better.
Hey, Dick, welcome Dickson Despommier.
Okay, Vince.
Dickson is at home, which is where I am, because it's snowing like crazy here up in the Northeast.
All true. This is very true.
And Dickson's on his cell phone, as you can tell, but actually doesn't sound so bad. But we are dedicated to getting out of TWiV, so that's why we're doing this. However, in western Massachusetts, where Alan Dove is, I hear it's raining. Hey, Alan.
It is. It's we've gotten a little bit of snow, and none of it has stuck.
Why is that? Is it always warmer there?
Well, no, it's supposed to be colder here, and we've got, every third truck here is a snowplow, and we keep not getting any snow. And, down in like Maryland, where they have no snowplows, they get dumped on. So it's...
Well, you see, you're too prepared. That's the problem.
That is the problem. It's like people remember their umbrellas, and so it doesn't rain.
And finally, from Florida, what's the weather down there, Rich?
Well, we have... clear blue skies and 55 degrees.
Jeez.
That's enough discussion. That's enough about the weather.
I hate it.
I mean,
this has been another TWiV.
This is really cold.
Hey, Rich, did you ever shovel snow, Rich?
Listen, I lived in Buffalo for 12 years.
Oh, that's right.
Oh my God!
Oh, you had your fill.
So I did my time. I know about snow.
Okay, got it.
Actually, I love it. I mean, I don't mind staying home.
Hey, if it's going to be winter, you may as well have snow.
I don't mind snow. It doesn't bother me. It's this happens to be some very wet and heavy snow. It's hard to shovel.
It's true. Actually, one person lost their life in Central Park yesterday because of that.
Is that right? Just walking, or
a branch of a tree broke because of the weight of the snow and killed him.
Yeah, it's very wet snow. That's too bad. Well, it's either that or the rabid raccoons. In Central Park.
The world is a different place today, isn't it?
It is. It's really dangerous. Well, today we have an all-email episode, and I got to tell you, this is a terrific episode driven by our listeners. Great questions, as you'll see. So let's dive right into it. And anybody can chime in anytime with comments or questions or answers about these questions. The first one is from Sheldon, who writes, I really like your podcast. Patterson, in a paper that he sends to us, links maternal infection to neurological damage to the fetus. A search for a vaccine in the article shows him favoring the vaccination program that makes rubella uncommon. At the end, he seems to be opposing flu vaccination because vaccination activates the immune system, which damages the fetus. If I understand it correctly, it's the number of antigens that count. Seasonal flu has 3 and the monovalent H1N1 has 1. How many antigens would a pregnant woman be exposed to during pregnancy? If it's a lot, then 3 or 4 more shouldn't make any difference. Thanks, Sheldon.
I would agree.
You think it's not good to be infected during pregnancy?
Well, it's never good to be infected by anything, even if you're not pregnant. But it's very difficult for things to cross the blood-brain barrier, but viruses manage to do it because they hitchhike across with macrophages that actually bring those things in. But what happens to them afterwards, though, because they get killed, right?
Well, what he's talking about in this article is the mother is infected, but then the fetus gets harmed because of cytokines that cross into the fetus, right? So he cites a number of epidemiological studies which suggest for both schizophrenia and autism, there's an increased rate in mothers who had infections during pregnancy. So actually this man who is, he's a professor at Caltech, he has a mouse model where he infects mice with influenza, and then he looks at their brains and he sees neurological differences, both behaviorally and structurally compared with uninfected mice. And he says, he concludes there's a link between the immune system and schizophrenia, but we don't know what it is. So he thinks it's cytokines. It's not actually virus crossing over. And so he says, rubella vaccine is great because it prevents a pregnant woman from getting infected. But at the end, this is the interesting thing, which I think Sheldon is referring to. Finally, I want to ask a question that's come up in the literature in the last few years. Should we really be promoting universal maternal vaccination? So he says, should we be giving flu vaccine to pregnant women?
I seem to recall that there was on the questionnaire for the flu vaccine a question about being pregnant. I'm not. Is that a contraindication ordinarily?
I think you're right. There is something on the form. I don't know that it's a contraindication.
It raises a larger question, though. What happens when there is a flu epidemic, as there is, and you're pregnant and you get infected instead?
Yeah, right.
Well, I think it's not good if you get...
if you don't on this one, it sounds like.
I think that's the bottom line, Dick, yeah.
I was just going to ask, there are lots of epidemiological studies that track offspring that occurred during and then you can fill in that blank, during starvation events like they had for the Dutch famine, and epidemics of, let's say, the flu or even measles. And there must be some good studies on this as to whether or not people who were immunized before those epidemics were better off or not after them. There has to be some hard data on this. We've done enough of those studies now to put it all together, I would think.
Yeah, he didn't mention any in the article. He did mention some studies where they looked at schizophrenia and autism born to women with or without infections, but not immunization.
Right. The CDC recommends that pregnant women get the flu shot. I'm looking at the H1N1 information right now. They do recommend that pregnant women get the shot. They recommend that they don't get the live flu mist.
Okay.
Right.
Which makes sense.
Well, yeah, I think the practical issue that Dick brought up makes perfect sense. Which would you rather have, the vaccine or the flu or the disease? Right. And I think the answer to that's pretty clear. However, this is an interesting concept. It's worth thinking about at least.
I think the studies haven't been done, but I don't think it's a matter of the number of antigens, because I think when you're pregnant, just as when you're at any stage of life, you get a lot of infections, right?
Yeah, right. You're just walking around, you're encountering stuff all the time.
Yeah.
Hell yes.
So
that's what your liver is for.
The part of this article that I like best, I didn't read it in any detail. I just kind of looked through it. But the thing I like best is he's got this handy guide to deciphering mouse psychology.
This is great.
You'll have to email it to the rest of us.
He's got 8 pictures of mice, and they're all exactly the same. It's just a mouse, right? And, each one is captioned, and one says anger, another says fear, surprise.
Oh, that's perfect. That's beautiful. Yeah, it's the same picture.
Right.
Depression, rapture.
That needs to be made into a poster.
Absolutely.
That is really funny. I missed that. I saw pictures of mice and I just skimmed over it. It's actually a good article. I will post a link, of course. But I didn't know this of his work, where he does infect mice with flu and then he does some fluorescence of the brain and sees structurally different things going on. So it's quite striking, I think. And so something that I had not come across my radar before anyway. So thanks, Sheldon.
Everything is connected, that's for sure.
It is, yeah. Anyway, the cover of this article has a nice painting by, is it Hieronymus Bosch? Yes.
Hieronymus Bosch, yes. Oh, yeah. I love his work.
Detail from the Temptation of St. Anthony. Lovely stuff. Okay, I wonder if he had an infection.
Well, he was certainly crazy.
He was crazy, huh?
He was 3 standard deviations from the mean. That's for sure.
I'm not sure I would want to have lived in his brain.
A lot of infectious diseases at that time in the 1500s, right?
No question.
I'm almost done reading Rats, Lice, and History. Was that it?
Yep.
I mean, people were just filthy. He said the streets smelled horrible. Everyone was filthy and infected. The spaces between brownstones in New York were made to dump your excrement out the window.
Of course.
You know,
they learned that from the Romans.
Unbelievable.
Based on one apartment I lived in New York, I would say that has not entirely changed.
Well, at the end, when we have our picks of the week, please remind me of a movie that I want to talk about.
Okay, we'll remind you of a movie.
The Devils and the Doctors. We'll talk about that.
Next one is from Andrew. I would like to know that I would like you to know that I enjoy TWiV and the new TWiP very much. Thank you for taking time to put it together. I was curious if it is possible to use gene therapy to insert genes into the major histocompatibility complex that could encode protein expressed on T cells for a certain virus. That way, it would be possible for you to have a wide variety of immunology to a number of viruses. The great part, the vector for the genetic material could be a virus. You will have to excuse any misused words. I'm not familiar with all the specific components of the immune system or gene therapy, but I am trying to learn. I guess I was just curious if it was possible. I'm a biology undergraduate with an emphasis in medical technology, hoping to get into a pharmacology program.
That's a deep question for an undergraduate.
So it's like he wants to. I'm trying to get my head around this. He wants to engineer virus-specific T cells. Is that the idea?
Sounds like it, yeah. I think the idea of putting the gene in the MHC is, I think he wants it to be presented, right?
Right. So, I mean, there have been, not really made by genetic engineering, but certainly there have been antigen-specific T cells made that are used in immunological studies that are, stuck into mice or whatever to study the behavior of those kind of cells in the absence of a background. So the idea of making an antigen-specific T cell is not an impractical idea.
Yeah, in fact, you remember we did this story on prostate cancer vaccines a while ago, and one of the goals of that is to immunize and make T cells specific for the antigen. Mm-hmm. I mean, I don't think you can co-express it so that it gets presented. See, he wants to put it right in the MHC,
right?
But I don't think you can do that just by putting it into the complex itself. It just doesn't work that way, right?
No, it doesn't work that way. After reading through this, my sort of bottom line on it was that, technically something like this may be possible, but nature does a better job just when you vaccinate somebody. You get the right cells, you get the right antigens, all that kind of stuff.
Yeah.
So let nature.
Very few exceptions.
Now there may be circumstances where something like this would. Where tweaking it in this fashion would be desirable, but nature does a pretty good job of this. I was trying to think, how are we going to get the right T cells? How are we going to get it just right? We got to make a different one for each virus. Blah 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 blah. Heck with it. Just vaccinate them.
Well, you can express viral antigens in vectors in a way that they stimulate T cells,
right?
And that's one of the AIDS vaccines. It does that, and you can use a viral vector, as you say. So. What you're suggesting is a good idea. It's just done in a slightly different way than you're suggesting it.
Right.
Okay, Jivan writes, love the show. Just a quick comment about the doctor you mentioned who recommended glasses as a means of protecting yourself from being infected by influenza. If influenza can infect the body via mucous membranes, such as the eyes, a pair of close-fitting glasses could protect one from direct transmission of the virus, such as a sneeze. Wikipedia says the three methods are direct, aerosol, and hand-to-mouth. Is it so silly, then, to recommend glasses that, in combination with a face mask, could stop all three methods of transmission? Have I got the wrong end of the stick? Keep up the good work. You're pedant. So we made fun of this several months ago. Someone suggested wearing glasses to prevent influenza. So is that really such a bad idea after all?
This isn't the only time I've heard somebody, in fact, another doctor, suggest that influenza could be transmitted through the eye. But I looked it up. I can't find any literature on this. Do you know of any, Vince?
I don't know of any that goes through the eye. I mean, it's theoretically possible because it's a mucous membrane, right?
Right. Yes. There's only one. There's a parasite called Trypanosoma cruzi, which is a vector-borne disease. The insect actually defecates the organism on your skin next to your eye and then you rub it into your eye, and that's how you catch it. But as far as I know, no one. I mean, if someone sneezed in my face, I would immediately try to wipe everything off. But I don't think I'd wipe them into my eyes. But I might. I might. It could happen. But you go to your mouth and your eyes so often with your fingers that you shake hands with somebody with influenza. The next thing you know, you've got it.
Yeah, I think the issue is that typical eyeglasses, I mean, like the ones I'm wearing right now, there's just an enormous amount of space around the edges.
Of course.
And, if I'm working in the kitchen and I splash something, I frequently get it into my eyes, even with the glasses on. Right. So in terms of just protecting the eyes, the glasses are not doing a great job. That's why there are safety goggles and safety glasses. But I think you're much better off with something like regular hand washing and possibly masks. I mean, that's something we've talked about too.
Or immunization.
Immunization,
most of the masks,
yes.
Most of the cheap masks don't work, so you'd have to spend a lot of money to really protect yourself.
Well, but that's something we've kind of gone back and forth about. There was just a study we talked about a couple of episodes ago that that addressed exactly that issue, that the cheap masks may actually help, though not as much as something like a $5 or $6 N95 mask.
And as far as I can tell, there's no good evidence that you're at great risk of getting influenza through your eye anyway.
Right.
I mean, it's
right.
So,
Al from Hawaii writes, "Aloha. I heard this on BBC News, but I could not see how it could possibly work." So basically, he sent us a study from BBC where they're going to filter blood, which is going to be given to children under 13 to remove potential prions so they won't contract Creutzfeldt-Jakob disease. So the BBC News says children under 13 undergoing a blood transfusion should be given specially filtered blood to cut the risk of fatal new variant CJ. So Al says, how is this going to work? Also, besides cannibalism and new variant CJ, D could be acquired from consuming BSE-infected beef neural tissue. So we stopped eating each other out here in the Pacific Islands, and the world...
We need to hear that.
Yes, and the world no longer consumes brain and spinal tissue.
That's what they tell the tourists anyway.
That's true. I saw that movie.
Aren't there nerves, neural tissue throughout all meat? I was wondering if TWiV thinks both of these prevention strategies are really likely to be effective in the long run, and if so, why? I'm confused. So I looked this up. They have made a company called ProMedic has made a affinity capture device that you pass the serum through, which it's an affinity ligand for the prion protein. I couldn't find what it was in any paper that I looked at. But they say it pulls out the prion specifically, and you pass it through it.
Really?
So they want. I think they want to sell this to the British government to use it for young kids who have not eaten the contaminated beef because they're young, right, under 13, and they don't want to get it from transfusion.
Well, if it's voluntary surgery, give blood in advance before you have your surgery.
Yeah, you can get it from someone. Yeah.
I think some economist would need to do a cost-benefit analysis of this.
Yeah, absolutely.
We're trying to protect against a risk that we don't even know is quite real. And the estimate is that just doing this for kids, this filtration system would cost us, in the article they say, £70 million a year for the national health system in Britain. I'm sure that'd be great for the company, but does it make sense to spend that much money on this measure to prevent something that may not even really be an issue?
Yeah, it says in this article that there have only been 5 known cases of transmission of CJ through contaminated blood and blood products. So that's not a lot, right?
Right. And the other issue is you have to know what the percentage removal rate is for this device.
It's never going to be 100%, right?
It's never going to be 100%. So if they claim that it absolutely protects, they're lying. If it provides 99% protection, there's some real risk. It may be worth this much money, but we don't know that until somebody really analyzes it.
I'm not even sure you could tell in an animal model if it were protective, because if you'd have to use so many animals, it might not be feasible.
Right, and you can't PCR the protein, so the detection of it would be I guess you could run a prion contaminated sample through it, and then do mass spec on it or some very sensitive.
It's very hard, right? Yeah. I mean, that's the problem. I was looking for ways to detect this. There's actually an interesting paper of just February this year where they do a protein cyclic amplification to detect misfolded prion. Very interesting.
How do you do that? A protein cyclic amplification?
Well, the misfolded prion makes more misfolded prions, right?
Right.
Yeah.
And
it looks really like a PCR, but
with protein.
I see.
Yeah, with proteins. Fascinating.
So yeah, basically that's it. You depend on the propagation of the misfolding, and then you can detect it that way. I mean, eventually they're saying that this will be used to scan blood products. I mean, or may be used. But the company ProMedic, of course, wants all blood to be filtered through their device.
Well, of course,
because they want to sell it.
Now, my reaction was the same as Alan's. It's going to be an economic issue. We're talking about a high cost for a low risk. Very low risk, as far as I can understand.
I mean, it's my understanding that you've gotten the contaminated meat out of the British system now, right?
Right. The disease is on the decrease.
Because you removed the feeding of recycled beef to the...
Right.
And I would think you'd also want to look at possible cheaper alternatives. If you can affinity remove this thing, you should be able to affinity detect it. So you could test the blood to see if it's there, and then you don't have to filter it. You just weed out any blood that contains it.
I'd rather spend the £70 million on developing a good diagnostic.
Yes, which would be much more broadly useful
than filtering everything. It just seems... I don't think that's ever been done, right? With blood to filter it for anything.
Actually, it has.
What?
You want to hear it?
Yeah, sure.
It's called the Goldschmidt procedure for getting rid of schistosomes.
Oh.
And these are a bit larger than viruses, but you can redirect the umbilical circulation through a filtration device which will actually trap the adult worms after you give a drug called tartar emetic, which is a very dangerous drug to give. It's nephrotoxic, hepatotoxic, and cardiotoxic. But outside of that, it's perfectly fine. It's not neurotoxic, at least. And that irritates the hell out of the worms. They migrate out of the mesenteric venules into the embryonic circulation, which is still a remnant of the umbilical, onto the filter, and you actually remove the worms. Then you can just take the filter out, and you don't have to use any drugs. So that's the only example I can think of where an infectious agent has actually been filtered out on purpose. But it was invented at Cornell University while I was a postdoc over at Rockefeller, so I knew all about it.
Now, Al had another question here about
trivial facts, right, guys?
No, that's great stuff.
Yeah.
Al had another question here about the neural tissue in the meat. Isn't there neural tissue in the meat? And as Dick said, yeah, there is. But I think that, correct me if I'm wrong, but I think this is basically a numbers game. In the brain,
CNS, though. It's only
the brain and the CNS, the concentrations of prions are going to be really high because that's a high concentration of neural tissue. So you get rid of that, and you decrease the numbers.
I'm not sure it's been found in motor neurons, has it?
I don't think so.
But again, that could be a concentration issue.
I don't think the entire cow is affected. I think its nervous system, central nervous system, is affected, but the peripheral nervous system is intact.
Okay.
But I think it can be found in other tissues other than the brain and spinal cord.
You know what? Why don't we poll our listeners?
Well, I mean, that's why you can eat beef and potentially be infected because it's contaminated, right?
Right.
Well, yes and no. Yes and no. It depends on how clean the meat is. The slaughterhouse technology.
Well, that's it, exactly. I mean, if it's very dirty, the spinal cord is on the floor and the meat. Right. Exactly. So, but it's contaminated.
And on the ceiling and on the
so maybe it doesn't matter that there are nerves in the muscle. The muscle gets contaminated. Yeah.
Never heard that one.
But if you take out the source of contamination, which is the contaminated cows, then the meat supply should be clean. And the only source is people's blood who were infected but didn't know it, right?
Yep.
And we have to figure out how to get around that. But it's very rare, so it's a tough problem.
And getting rarer because since they understood what's going on and cleaned up the cattle industry and the feeding practices and that kind of stuff. It's been on the decline in the cattle and
Correct.
on the decline in people, as I understand it. So,
and in Papua New Guinea, kuru is almost non-existent. Yeah.
Right.
Well, here's the question: if your kid needed a transfusion, you're living in the UK, you have a kid under 13 needs to have surgery, and you can't get someone to donate blood. You had the opportunity to have it filtered through this filter. Would you do it?
Thanks, Vince.
There's a dead silence at the
That's
end.
great. Yeah, dead silence. Well, actually, that's an interesting approach to the whole thing. Listen, we can give you the regular blood, or we can give you this stuff that we filtered.
That's right, exactly.
For
right.
this many dollars more.
Yeah.
And the public health service.
You know what I would do? I would poll all of my relatives first for that right type of blood, and I would probably trust that more than I would trust anything else.
Yes.
Well, I was saying if there's a situation where you can't get it from relatives, right?
Yeah,
Well,
that's right.
comes back to the economic issue again. How much is it going to cost me? Right?
Yeah, I mean, look, if
what you're describing is the normal situation now.
Right.
Yeah, you're saying if you go in a hospital, you're in a car accident, and they've got to give you 2 pints of blood now, or you're going to die. They just give it to you.
if they asked me if I want it filtered, I'd get it filtered.
I think I'm not conscious. I can't answer any questions.
If the only downside is extra cost, I'll have it filtered.
Yeah,
That's fine.
I'll admit, I would have it filtered, but I would not be in favor of having my, in Britain, you've got a healthcare system that's government financed,
Right.
like it or not. But I wouldn't be, I'm not going to get political about this, but I wouldn't be in favor of a taxpayer financed system paying for something like this unless somebody's actually done the analysis and shown that it is cost effective to do it this way.
Right. I agree with that.
But on my own dime with my own kid, I'd probably
Yeah.
in the UK, I'd probably go with the filter.
Interesting.
Okay, Al, thanks for that. That's a good question. They're all good.
Yeah.
Before we go on, we have a bunch more. What's that, Al? I mean, Al. What's that, Dick? Sorry.
No, I said that kept us busy for a while.
Well, prions.
We covered about 5 different subjects in that one question.
It's okay.
That was good.
It's the whole. We're just shooting the breeze, and prions are very interesting.
Yeah,
it's the good stuff.
Absolutely.
Before we go on, let's acknowledge the support of TWiV by Data Robotics, the makers of Drobo, the backup and working solution for your computer. Drobo, Data Robotics makes these wonderful little appliances where you can hook them up to your computer and store information on them. And they have slots to put drives in, and you plug them into your computer, copy things over, and it's safe because the data is stored redundantly. If one of the drives fails, you can pull it out while it's running and pop a new one in, and it'll be restored. It's a beautiful solution. And now that I'm sitting here at home, I see my 2 Drobos stacked up here with all the old TWiVs on them and all the other miscellaneous files that I don't want to lose. And once I copy it to that machine, I'm confident that nothing's going to happen to it. So Drobo is supporting TWiV for a couple of months, and the neat thing they're doing is they've given us 3 units to give away. So head on over to twiv.tv/drobo-contest to find out the rules to win one. This is the last week for the first giveaway. We're giving away one a month. And at the end of this week, we'll pick a winner. And then the following week, we'll devise some new contest, which does not involve Twitter, for those of you that don't use that. And then there'll be 2 more to give away. And it's a good way to raise awareness about viruses and Drobo, which is a great backup solution and also a great solution for working with files. They have new models that are very fast now and work very well for not only securing your data, but being able to work with it as well. So head on over to TWiV and check that out. You can also go over to drobo.com/TWiV to learn more about these terrific appliances. And we thank Drobo for their support of This Week in Virology. Our next email is from our friend Ricardo, a professor of microbiology in Portugal. And he sent a link, 2 links. Did you see the news about the second viral disease to be extinct in the next year or so? Rinderpest A bovine disease. Know about rinderpest, Dick? This is I actually was not aware that it's almost eradicated. Anybody know about this?
I hadn't heard about this either.
I know about the disease, but I didn't realize that it was that close to becoming a relic.
So what's the disease like, Dick? Do you know?
It's mostly an East African disease of animals, mostly hoofed animals. I believe it's vector-borne, and there's very little else known about it, except that I believe that there is a good vaccine for it. So I think that, at least in domestic cattle, you can keep it out pretty well. It's one of those diseases that they're examining out in Plum Island because it's a veterinarily important disease, and as a result, there's been a lot of interest in controlling it, and apparently they've been doing a very good job.
So, according to the Merck Veterinary Manual, it is a disease of cloven-hoofed animals, characterized by fever, necrotic stomatitis, gastroenteritis, lymphoid necrosis, and high mortality. In epidemic form, it is the most lethal plague known in cattle.
Exactly.
And it's spread by nasal secretions, not vector-borne. And it's a member...
Flies can transmit it from cow to cow, that's what I know.
Okay.
You know, the flies that hover around them.
Apparently, it's had a huge impact on agriculture over the years from
right.
when it was first discovered. This is actually a virus related to measles virus. And in fact, some people believe that measles was a zoonosis that originated from rinderpest in cattle. Maybe 15,000 years ago when humans began to domesticate cattle, right, to eat them. They started infecting themselves with the cattle virus, which was rinderpest, and it became measles.
I guess the transitional form was measles.
Measles. Yeah. It also affects other animals besides cow, buffalo, giraffes, and other animals that I can't pronounce. Wildebeest.
Well, I can tell this is a live vaccine that's tissue culture attenuated in the old-fashioned way. That's what I get out of it anyway. That's very effective.
Yeah, apparently it's a great vaccine, and it's been really effective at reducing the disease. So they say here at one article I'm looking at that Ricardo sent, and within 18 months it's expected to be eradicated.
So how is it that we're able to deliver an injected animal vaccine to all the cattle in East Africa, but we can't wipe out polio?
What a good question. Would you have an answer?
I have a provisional answer, which is that people have fewer misgivings about having their cattle vaccinated by foreigners than they do about having their kids vaccinated.
It is interesting, isn't it?
The other thing that's going to be interesting here to see if you've got reintroduction of the virus from the wild back into domestic cattle after they discontinue the vaccines because they consider this completely eradicated.
That's a good point. That's a good point.
Go away from the wild animals.
In order to consider it eradicated, it's going to have to be eradicated from the wild animals too, I assumed.
That's impossible. That's totally impossible.
In which case, it's not eradicated.
Well, I guess what they mean...
You're right.
I hope what they mean is that they're going to eliminate it from the domestic cattle.
I think that's
but if it's in... You're right. You can't vaccinate every gazelle in Africa, and if you don't, then...
Good Lord, no.
Then this is still going to be circulating.
Good point.
I believe that's true.
I think that this is not really eradication.
No.
As it is for polio, where the only reservoir is humans, and once you get rid of it there, then it's eradicated.
Yeah, except that it can linger for about 3 years in the ooze, right?
Yeah, well, that's another problem, right? Actually, in this article, Alan, you may see this, the last paragraph of this news.com article.
Yes.
Could eradication be delayed, similar to polio? Well, I think, given this consideration, that if they get rid of it in cattle, it's just a matter of time before it pops back in from a wild
I saw something, I thought I saw something in one of these articles about them being close at one point and then it coming back, or am I thinking about another one of these articles?
That's true for everything, I think.
Yeah, he does say this at the end of this article. He said he's making an analogy with polio eradication. where we got close and then never got to the end. I would say the same thing has happened with rinderpest. The last outbreak was 2001, but let's say in the 5 years previous, we did have the trickling of evidence because the vaccine was not being applied correctly or control programs did not occur. This led to a few outbreaks.
Right.
So I don't know. I think they're a little bit optimistic here.
Yeah, and I think
and they're also they're focusing on the domestic cattle rather than the wildlife.
Yeah, I think the use of the term eradication is inappropriate here.
I would agree.
You think that once it's declared eradicated from cattle, that they will continue to immunize cattle?
No.
I wouldn't. If I didn't have to spend the money, I wouldn't do it.
Well, then they're asking for trouble.
Yeah, I mean, we can't even keep vaccination campaigns for polio going after it's been quote unquote eliminated from a country.
Right.
That's what happened in the Dominican Republic a few years ago. They declared it eradicated in the Western Hemisphere. Everybody turned off the lights and went home, and then
we have a rich history of doing that.
Yes.
All right. The next email is from Zach. Hi, I just wanted to let you know that the 365 Days of Astronomy, which is a podcast, will be going for a second year, and they are looking for people to do podcasts. Not quite sure how to talk about virology in an astronomy podcast, but if you could think of something, it might be a good way to bring more people to listen to TWiV. Hey, this reminds me that in one of my classes, before I started, I asked the class, "Do you think there are viruses on the moon?" And we had a great discussion, actually. So the idea of viruses or other things in space is very intriguing.
Do you remember the movie 2010? Not 2001, A Space Odyssey, but 2010, the follow-up movie?
No.
Well, the monolith, you know that big monolith that attracted everybody, and then it ended up in Io. Remember they said, keep your hands off, it was either Europa or Io. I think maybe it was Europa. This monolith then replicated like a virus. They actually showed it happening. You might want to download that last scene and put it up on TWiV as an example of a viral replication cycle.
Was that made by Stanley Kubrick?
Yeah.
The sequel?
Yeah. And Arthur C. Clarke wrote the book. So,
2000. All right, Alan, look it up, will you?
It was 2010.
Yeah, I'm working on it.
All right.
They've got.
And at the end of the movie, it showed this. I even forget who the actors were, but they were all famous actors. It was a Russian team and a U.S. team of space, astronauts that flew out now to see what was happening. And I don't think Io was the moon. I think it was Europa.
Oh, Io, the moon of Jupiter, you mean?
Yeah.
Okay.
Well, the one that the voice told everyone to stay away from. You see, there's this mystery about you're going to have a wonderful existence and everything is going to be peachy keen as long as you keep your hands off of. And then I believe that the name of that moon that they said to do it with was Europa.
Okay.
And then it shows Europa, and the monolith goes into the moon and then starts to eat it, I guess, or something. Who knows? It was a very surrealistic scene, but it looked like a virus replicating inside of a cell.
This sequel was not 2010 was not directed by Kubrick. It was
not by Kubrick. No,
Peter Hyams.
Okay, take all that back.
Yeah, it was.
But the book was written by Arthur C. Clarke.
Yes, the book was by Arthur C. Clarke. Movie was released in 1984. I remember when it came out. I never saw it, but I had seen 2001.
Visually interesting, but as far-fetched as 2001, of course.
Zach continues. Professor Pogue at OSU has an interesting podcast from a class he gave on life in the universe this fall, and he sent us a link to it. It's the class website for Astronomy 141, Life in the Universe. Ohio State University's got a couple of podcasts of his lectures there, including some on life in the universe, so check that out. Thanks for that, Zach. And Jim writes he sent us a link to an educational game put out by the Federation of American Scientists. It's called Immune Attack, which will plunge 7th to 12th graders into the microscopic world of immune system proteins and cells, a three-dimensional video game. So I went to download it, and it is a PC-only game, Windows only.
Boo.
I didn't try. Man. gotten on top of this.
And I haven't booted into. I could boot into Windows on my Mac, but I haven't done it yet. So we'll post the link to that, and someone can try it out and let us know. I was going to run it.
Put your son on this. Isn't he into this?
Yeah.
Stuff?
Yeah, I told him while I'm on the podcast here, he's got to cut back on the bandwidth, because he's up there gaming away. Yeah, he's into this. I'll put him on it. All right. Ibrahim writes, I was reading a review. One of the authors is my supervisor. Well, I guess you should read it then, right? Where I found this paragraph, which I need your comment on. The occurrence of oseltamivir-resistant seasonal influenza virus did not appear to be directly related to the overuse of oseltamivir. 67% of H1N1 strains in Norway are oseltamivir resistant, though the drug is rarely used there, whereas in Japan, which has the world's highest per capita oseltamivir use, seasonal H1N1 resistant rates are only 3%. Okay, so this is a well-known issue. And let me quote from a very nice paper by Eddie Holmes. He calls this genetic hitchhiking. There's been a dramatic rise in oseltamivir resistance in H1N1 seasonal viruses circulating in Europe, which was caused by a histidine to tyrosine mutation at position 275 in the neuraminidase protein. Because these drugs are not widely used in European populations, linkage to another beneficial mutation seems the most probable explanation for the rise of viruses with that mutation, although this needs to be formally assessed. So basically, it's thought that there was another change in the virus which helped it transmit better, but it wasn't so viable, if you will, and this second change, 275, then was selected for, and that happened to confer resistance to oseltamivir. And he also cites another example with amantadine resistance in H3N2 virus is the same idea. It's not actually selected by use of the drug, but by this genetic hitchhiking. So it's quite common with flu, apparently. So sometimes you get selection by the use of drugs, but not always.
Malaria seems to work in that direction. The higher the use of any of those commonly used drugs like chloroquine or some of the other derivatives of chloroquine, the next thing you know, there's a whole bunch of resistance going up because these are across-the-counter drugs in a lot of places, and they overuse them.
Right.
And the organism, of course, has a mutation rate that's keeping up with the evolution of drug development.
Yeah, I think that with flu that happens occasionally, but there are cases where it's independent of drug use.
Right, the mutation that confers the resistance confers some other advantage in a different context.
Exactly. So there you go, Ibrahim. Peter from Korea sent us a link to a podcast where Dr. Moria Gunn sits down with the president of Global Vaccines and Infectious Diseases of Merck to talk about new vaccines on the market. Okay. And I found this very interesting, not just because of the vaccine, but this is from a podcast called Tech Nation, which is hosted by Dr. Moira Gunn. She has conducted over 2,000 interviews with space pioneers, cyber novelists, venture capitalists, and genetic researchers. She's a former NASA scientist, and she was the first woman to get a PhD in mechanical engineering from Purdue. So here's someone who apparently has a second career in radio and is a bona fide scientist. So that's interesting in itself.
This is an NPR is involved in this. Is that right? Public
radio, yeah, Tech Nation, which I'd never heard of before. Anybody else hear of this?
Nope.
All right.
Actually, they have the website where this is on, there's a whole series of such programs, but this looks very good, and I'm planning to listen to more of them. Had anyone listened to the vaccine conversation?
I did.
Yeah, how was it?
It was good. They discussed three. Well, they're all Merck vaccines. It's a little bit. Well, it's not self-serving. Heck, it's about what they do.
Yeah.
And just to point out, Merck is probably the leading manufacturer of vaccines.
Right.
I mean, I have no connection with the company, but for years they dominated that market.
Right. And I'm glad somebody's doing it.
Yeah.
So they discussed. Actually, it's a nice discussion because it's three very different vaccines. They discussed Gardasil, which is virus-like particles, and Zostavac, which is a live attenuated varicella zoster vaccine, and Rototec, which is for rotavirus gastroenteritis disease, which is, that to me is the most interesting vaccine, because that one is so the rotaviruses are double-stranded RNA viruses with a segmented genome, so they can do reassortment the same way influenza can, and so you can make reassortant viruses, and the Rototec vaccine is essentially attenuated by virtue of being a reassortant between several human serotypes and a couple of bovine serotypes. I don't understand exactly what the ultimate viruses look like, but what I get out of this is that part of the attenuation process is to make reassortants that have bovine genes in them, and they don't work in humans, as they aren't as pathogenic in humans. So, interesting vaccines and a very understandable conversation.
Now, is Rototec made the same way? There was RotaShield, which was the one that was taken off the market, because I remember when Rototec came out, and there was this concern that it might have some of the same problems.
Right, and I don't know if they're made the same way or not.
Okay.
I'd have to look it up.
Yeah, it is a reassortant also.
RotaShield is?
Okay.
Yeah.
Okay.
It's a reassortant between human and rhesus rotavirus, yeah. Whether it's the same or not, it probably is different enough so that it wasn't associated with the intussusception.
Right.
The intestinal issues where the little intestine kind of goes into, at the place where the small intestine and the large intestine bifurcate.
The junction.
Yeah, the junction. Yeah. The small one drives up into the large one. And
I didn't realize that Merck had developed the Zostavac. I mean, that vaccine, or at least the concept of that vaccine, if I understand it correctly, was originally developed in Japan.
That's right.
It had been used there for a long time. I didn't realize that Merck, from this, I gather that Merck has made its own thing, which I was.
Merck has a big office in Japan, too, so maybe it's the Merck office in Japan that did it.
I think they took the strain from the Japanese scientists and, pushed it further in some way.
Okay.
But I do believe they used the original strain that they had used.
And it's worth pointing out that the vaccine is exactly the same thing as is, if I understand it correctly, that is given to children as a chickenpox vaccine that they give to people over 60 at a higher dose.
Yeah, right.
For shingles.
That's right.
Okay. The idea is it's basically a big booster, because the reason you get shingles is that you've got this virus latently hanging out in your neurons, and as you get to be an old codger like certain people I know, and
I resemble that remark.
Your immune system starts to sort of get a little tired. A booster helps.
I've had shingles.
You've had shingles?
Shingles ain't no fun, kids.
So I'm told.
The point, Rich, is that because the virus is latent, you don't have a boost of your immune system, right?
Right.
So it wanes.
And your immune system kind of, when you get older, kind of wanes some, and so this booster with this vaccine helps you out.
Well, I'm going to listen to that. I think this Moira sounds very interesting here. So
it's a nicely done interview.
Good. Thanks, Peter.
Good deal.
Frank writes, always engaging, keep it up. In TWiV 63, you discussed human viral infections, which are asymptomatic. Various sources commonly refer to the human body being composed of more bacterial cells than human cells. Some claim 10 times. I assume that each of these bacteria are susceptible to virus. If so, are any human health problems associated with the disruption of these valuable human resident bacteria?
Goodness gracious, what a question.
This is a good question. Whether or not killing of the bacteria by phage is an issue, we don't know. But what we do know is that phages transfer genes among bacteria, and that can make them more virulent. So the one that comes to mind, this E. coli 0157,
sure,
you can get from hamburger that's not cooked properly, right?
Right.
Yep.
So that has a toxin in the bacterium that was brought there originally by a phage. It's called Shiga toxin. So that's just one example of a phage that can make it more pathogenic. Now,
doesn't cholera have something similar going on?
Yeah, sure.
Yeah.
Of course it does.
Exactly. Cholera toxin is on a phage, right?
Yeah.
And diphtheria bacteria too.
His question, though, is different, whether destroying the bacteria is an issue.
Yeah.
And I think,
exactly.
I think we don't know. I looked and there isn't any work yet to really address that. But I'll bet there's
actually, I went to a talk on this just a few months ago. There's been some work on using these genomic sequencing systems to sequence the gut microbiome. And there has been some interesting preliminary findings about how your changes in your gut microbiome can affect overall condition and health.
I'm sure we're going to hear more about this.
Yeah, this wouldn't surprise me a bit.
Yeah.
It's amazing people haven't paid attention to this before because we are all walking ecosystems. And I read an interesting review of a book recently that started to actually describe all of the ecological niches that we present for our microbial flora and fauna. And it changes over time.
Well, the sequencing techniques that are available now make a lot of these studies possible. I mean,
that was
previously it was all dependent on what you can culture, and you can't culture.
Yeah, that's right. You're right. You are right, and it makes us appreciate who we are even more. I think I gave a lecture yesterday, just as an aside note, and told everybody in the room. I said, "Did you know that when you're born, you're totally sterile? You have no microbes whatsoever," and no one knew that. I said, that's why little babies cry when they start to grow up, because they start to acquire all these things and they get lots of gas and everything else.
Dick, I thought you were retired.
Oh, I'm still teaching. I don't retire from that ever. That will always go on. But, it's amazing how many women did not know that their fetuses were totally sterile. I said, imagine them being infected. I said, you wouldn't want to be around when the amniotic fluid burst open because of all the microbial byproducts that might be there. But the second thing is, of course, the fetus wouldn't survive it.
Right.
Can't stand the competition.
Yeah, in fact, one of the things that came out of the genomic sequencing work was that you can actually trace microbiomes in families from people's early environment when they're growing up.
Yeah. It's fascinating stuff.
Okay, Rick writes, "Happy holidays, guys." You can see how old it is.
Backlog.
Yeah, you're behind.
That's an old letter. That's got to be an old letter.
Oh, no. I'll tell you what. He was wishing us a happy Valentine's Day. That's clear.
Or Easter, perhaps.
Yes.
I have a question about all the H1N1 vaccine recalls. The recalls seem to be based on loss of effectiveness or weakness over time. If the vaccine is weak, is there a greater risk of H1N1 becoming resistant to the vaccine?
Well, viruses don't really become resistant to vaccines, do they?
I think what he means is,
I
think he means, selecting for antigenic variants.
Yeah, I was thinking
that the vaccine doesn't affect.
Yeah, I was thinking, in fact, the opposite might be true. If the vaccine is weak, then there's less pressure on the virus to drift towards something that is an antigenic variant. I mean, this is just in the hypothetical realm.
Right. Yeah, because what the vaccine does is prime the immune system to see this virus, and if it primes the immune system inadequately, then the virus comes in and doesn't have to deal with the vaccinated immune system.
Exactly.
Yeah, my feeling is it's no different from, when you're fully immune, or let's say you have an infection, you get a great immune response, you can still select for antigenic variants that evade the antibodies.
Right, which is why the immune system... Right, and that's why the immune system does its own selection as well with hypermutation.
So I think the answer is no. If the vaccine is low potency, you have a bad immune response, you won't be protected. That's about it.
It's essentially the same situation as vaccines that have to be delivered in a dose and booster regimen. If you only had one, and then you get the infection, you're still at least partially immunized, but it doesn't really affect.
Yep.
Yeah.
All right, Mark writes, I just finished listening to your XMRV podcast and also enjoyed the panel discussion you participated in on futures in biotech. Since there are a few pathogens that we know about that cause cancer, either retroviruses whose insertion carries an oncogene or ones that activate a dormant oncogene. I was wondering if there has been any systematic effort to look for signs of other retroviruses in other tumor types.
I don't know about a systematic effort, but
there are other viruses that cause cancer. Oh, I just saw your annotation down at the bottom, Vince. Yes, there are other.
Sure. Oh. That's okay.
Yeah, just pointing out that it's cancer is not a strictly retroviral thing in the viral world. Hepatitis B, hepatitis C, papillovirus,
memory tumor viruses.
I think basically when you have a tumor, you look for you do epidemiology to try and get an idea of what's going on. Of course, now we have DNA-based epidemiology.
Right, in the Tumor Genome Project.
Right
now.
So I think what we want to emphasize to Mark is that it goes beyond retroviruses. Viruses of all different families can cause tumors because the mechanisms are different.
Wait a minute, Alan. Did you say Tumor Genome Project?
I'm sorry, Cancer Genome Project.
Okay, so this is a real deal?
Yes, the Cancer Genome, I think that's what it's called. Just asking my friend Google now. Yes, the Cancer Genome Project. I mean, you've got all these high-throughput sequencing machines sitting around. You got to find something to do with them.
Right.
Sorry, I shouldn't have put it that way. But
is that like the infinite number of monkeys? Probably.
Since that genome-wide association study thing worked out so well, there, the Sanger Institute and the NIH and the usual suspects are collaborating on this. So if you go to cancer.gov, you can find some information aimed at a general audience. And the idea is they take particular types, as Rich, I think, as you probably realized, and what's lying behind your question is that this would be an enormously ambitious project, right?
Mm-hmm. But it addresses Mark's question of whether there's a systematic effort,
and the answer is yes.
Signs of anything. I mean, he says other retroviruses, but if you got a big sequencing project going on, and it's appropriately done, and there are other viruses associated with specific tumors, that sort of project might turn that up.
Exactly, and that is exactly the sort of thing they're taking. I think pancreatic cancer and lung cancer are a couple of the first ones that they're looking at, and they're just going to the pathology labs and taking all these biopsies and sequencing as many things as they can and looking at the distribution of different mutations that you got in different patients. And finding, of course, some patients, you're going to have clustering around certain types of mutations that will occur, and you can try and find out which ones occurred earlier and which ones occurred later as the disease progressed. But in the process, I think they're also looking at the retrovirus integration in there.
Sure. Well, that's interesting.
Yeah, it's an incredibly ambitious project. I hope it works out well, but I think it's a really neat approach.
Mark also asks if there's a podcast similar to TWiV focused on oncology research. The podcast.
There's no podcast similar to TWiV, excuse me.
Except for maybe TWiP, huh?
We stand alone here.
The podcasts I've seen before seem to be either patient-oriented or focused on the clinician. I haven't seen any like yours that bridge the gap between layperson and researcher. I'm a software engineer by training, and I develop bioinformatics applications for pharmaceutical and biotech companies. So I find your podcast particularly helpful in learning both about pathogens and the assays that you use to characterize viruses and evaluate vaccines. I don't know any, because he's right. They're mostly patient or clinician oriented. There are lots of podcasts by docs talking about, oncology and similar subjects, but very few basic science. Any of you guys know of any?
No, not me.
Anyway, our listeners, if you know of any, let us know, and we'll mention it for Mark. But I've always looked, and we seem to be filling a void, which is good. But other fields could welcome. If you remember, a physician wrote us a while ago about This Week in Cholesterol.
How about This Week in Dark Chocolate?
Well, that's a good one.
Yep.
My knowledge would fill the first 30 seconds, and then that would be it. But it would be an interesting show anyway.
Exactly.
Okay, shall we continue, gentlemen?
Sure.
Yes.
Richard writes, I'm still enjoying your podcasts, being a listener to both TWiV and TWiP. I'm a sewage treatment plant engineer, but have an interest in science and enjoy listening to podcasts such as yours. It has no real bearing on my work. I simply enjoy learning about such things. Although it may lead to a radical shift in career path, who can say for sure? You don't think it has no bearing.
You never know what might collect in the Schmutzdecke.
Yeah,
That's right.
I was thinking about the Schmutzdecke. I wonder if he heard that podcast.
I think he shouldn't say it has no bearing on his work. It may make him think in a different way, right?
Sure.
Yeah.
So these subtle things that happen. Anyway, he continues, I was looking into RNA interference used in genetics to study the blocking of genes. This appears to be making use of a viral defense mechanism that is in many types of cells, from plants through insects to people. This seems like it should stop some viral replication in its tracks, but clearly this does not happen. I wonder if you could discuss this topic and clarify what the virus does to avoid being stopped by this in the cell. I have a few ideas, such as blocking the cell from detecting double-stranded RNA, or by including its own enzymes to interfere with or replace the cell's own machinery. Unfortunately, I have no way to test these, so it would be nice if you could clear things up. Many thanks for the time you put into creating these podcasts.
Okay, maybe he is in the wrong job. That is an outstanding question from a
great question.
Yeah, right. He goes right to the heart of it.
Yes.
And yes, indeed, viruses do make things like proteins that bind double-stranded RNA to counteract the effects of these silencing mechanisms.
Yeah.
Yes.
Quite a few mechanisms are known for a wide range of viruses.
And they use the approaches exactly that you're mentioning in this letter. You've not only hit at one of the core questions in virus-host interactions, you've proposed some of the ways that it's dealt with. So Richard,
I like the part where he says,
"Give up your day job."
That's right.
He says, "I have no way to test these." I think that's interesting. It sounds to me like he wants to do an experiment.
Yeah.
It's probably all happening there in his Schmutzdecke right outside his office.
Absolutely.
So basically, go ahead.
Yeah. This is a really, really good letter.
He's Richard, I wanted to tell you, for every defense that the cell puts up against viruses, the virus always evolves to counter it, no matter what it is, right?
It's an ongoing tug of war.
I think we say this every episode, sorry.
Yep.
At what cost, you said last time, Alan, right?
That's
What's the cost? That's the only
I really like Richard's tagline on his letter.
Yeah, go ahead. Tell us.
Quote from Pablo Picasso says, "Computers are useless. They can only give you answers."
Yes.
What did he know?
That may have been true before the internet, but I think now it's a bit different, right? Because Picasso was pre-internet, right?
He was.
Yes.
I think you get
If he
plenty
could
of
only have lived long enough to sell his paintings on eBay, he wouldn't have said that.
Look, my computer gets me questions via TWiV, not just answers.
Yeah, but it's the people providing the questions. It's not your computer.
No, not right now, but someday.
Trouble is, the answers were never specific to the questions.
Okay, the next one is a praise letter. So if you want some praise, guys, it's from Jenny, who writes, I wonder whether we have entered a new chapter in the book of human knowledge sharing. I'm often working physically when I listen to my favorite science podcasters, TWiV, TWiP, and Mark Crislip. So your entertaining program is intertwined with raking leaves, loading wood, washing dishes, but not vacuuming. I hope that learning and moving can be merged in this decade through a more thoughtful use of tech. I love the unveiling of your subjects over the course of time. The interaction of the two of you is very important in peeling the layers. Both of you ask each other good questions, and your model is very important. Neither of you is afraid of appearing ignorant by either asking questions or not knowing an answer. Other aspects that you are modeling, the power of synergistic professional-personal friendship and the sublimation of professional rivalry that can humanize science and extend knowledge boundaries. For years as an RN, I've taught healthcare to people with a high school education, maybe not high school sometimes. The wedge or funnel of narrative is by far the best way of getting the information into their skulls. Stories, pictures, and practice, actually. Your stories are the best. Take good care of yourselves. Walk where you want to go this year. Eat more vegetables. Have fun in the physical world, and go get them in 2010. Your listener,
Wow.
Jenny.
Cool.
Hey, Jenny.
Nice.
By the way, Jenny, that's an excellent safety point for listeners. They should not listen to science podcasts while vacuuming.
This is true. While operating heavy machinery.
Well, no, specifically while vacuuming, because nature abhors a vacuum.
You know. I
really like. She obviously is somebody who appreciates people cooperating with each other rather than competing with each other, and that's something that we can use as much of as possible in science.
Yes.
Here.
There has to be some competition because there are not enough resources for everybody, unfortunately.
Unless you're retired.
But we need to cooperate as much as possible. It makes it more fun.
It should be friendly competition.
Yes.
That's right. As I like to put it to my students, I said, this is not a competition between all of you against each other. I said, I'll draw a line in the sand, and everybody can come on that side with me. Now you look back across, and what you're looking at are the diseases, and they're winning. It is a competition, but it's all of us against them. And so we need everybody. We can't do this alone. I don't care who gets the credit. I just want to see something get better for us. And that's the reason why I did what I did for however many years I did it.
Indeed.
And that's why you're doing.
And the students buy that.
You're still doing it.
Yeah, you're still doing it.
The students bought that because. Well, go ahead.
Well, I was just going to say the reason all of us are doing TWiV is for the big money we make from it, of course.
Well, we don't want to tell everybody that, do we?
It's out to
By the way, our next fundraiser is starting with TWiV 72. Right? Annual, I should say, annual.
Yes.
Eat more vegetables. Thank you, Jenny.
That's a great idea. We will do that.
Wayne writes, can you please discuss this science article on TWiV? And this is the Nature article on the bornavirus DNA in the mammalian genome, which we discussed on TWiV 65. But here's his question. I would like to better understand why viral DNA is kept in the mammal DNA. Does the viral DNA provide a survival advantage to the mammal? If not, how does the embedded viral DNA survive the process of evolution? Or is the virus continually incorporating its DNA into each generation? I think we talked about this last week, right?
Yeah, and this paper was actually a few episodes ago what originally started our conversation about bureaucratic evolution.
Yeah.
Yeah, and I'm in Alan's camp on this one. Okay,
yeah.
I think that sequences can hang around for a long time, for no particular reason. They don't because they're there. It doesn't necessarily mean that they have a function. I mean, even pox viruses carry around a lot of apparently broken genes. I don't know that anybody's done the appropriate phylogeny to know how long they've been there. But there's a lot of genes that are just kind of they broke some time ago, and there's no particular reason to get rid of them. So
it's just it's more effort to take them off the payroll than it is to just let them sit around.
Exactly, that's the bureaucratic nature of it.
See, Alan, you've already said two things now that contradicted themselves. Nature abhors a vacuum. That's number one.
Which was a joke.
No, but it's the truth. I think you can't create a vacuum in nature. Something will come to fill it. You extinct a species and something will replace it. And invasive species are very much a good example of that, right? But then I take another approach to this, and it's a philosophical one. If you're going to spend all that energy replicating your genome, and there are other things in it that take even more energy that are not your genome, why would you do it unless there was some selective advantage to it?
I think you're overestimating the amount of energy involved in replicating that versus the amount of energy it would take to have a system that would correctly excise the exogenous DNA.
Right.
Yeah, well,
remember, if you screw up the removal of this DNA, you're going to die.
Well, that's the point. How can you do the experiment? Right? You have to take a simpler organism like the virus that you just mentioned that has what you think are broken genes. You could probably remove that mechanically, in the laboratory, and then sew the organism back together again without those broken genes, and then see what happens in a cell culture medium and see if there are some advantages or disadvantages. But I don't know, has that been done? I mean, take the introns out of something and then just find out what happens.
Well, I mean, we do have some natural experiments like fugu, which I think we mentioned last time, which has virtually no transposons in its genome. It has very little waste DNA, and it seems to get by just fine.
Right.
So you can build a complex animal without all this junk DNA.
Right.
And it's not. I think that almost proves the point. Maybe not quite, but almost proves the point that it's not really necessary.
Hmm.
I also think of the quote junk DNA. Now I'm making this up entirely as blank tape. Sequences that you might be able to do with something, do something with sometime.
Right. That's another issue. You do probably want a certain amount of slack in the system that may do something useful at some point.
Well, then it would have a purpose then.
I've also wondered many times whether some of the stuff, and obviously this is part of the question, some of the stuff that we call junk, we just don't understand what it does. And in particular with DNA in the nucleus, I wonder if there aren't, some of it serves for structure rather than function.
Oh, absolutely. In fact,
structural considerations that we don't understand.
And certainly, if you do like the experiment Dick just outlined, if you're working with a virus and you take out the so-called inessential genes and you show that it grows in cell culture, all that means is that it's inessential in cell culture.
Right. Correct. No, I realize the limitations of the experiment, but I remember someone reducing the number of genes in E. coli from 3,000 some odd down to 350.
Right. And
given all the growth conditions on the outside that you've supplemented to make up for those genes that you've lost, you can still get something that looks like a bacteria.
Right.
How many genes are minimally necessary to have a self-replicating organism not dependent on another life system?
I think Craig Venter was doing that experiment at one point, wasn't he?
Yeah, that's right. So, it does get you right up against the philosophical wall of what life is. And we've actually tossed that one around a few times too.
Oh yes.
Yep. There's an interesting paper in Genome Research this month called Genome-Wide Discovery of Human Heart Enhancers. So they developed a software algorithm to search through the non-coding DNA. The coding DNA is only 1.5% of the human genome, right?
That's what I was going to follow up with.
So they say they can find stuff in the non-coding DNA which makes a heart cell and a muscle cell a muscle cell.
Oh.
So, enhancers can work at a distance. So that might be part of the story. So
maybe it's genetic punctuation rather than...
So, Dick, you would like this. I read a blog post about this article. And they liken this software to a fish finder. You use a fish finder to find the prize catches in the waters below. And like that, these guys have developed a computer tool to scan the DNA depths for the tiny switches important for cell determination. There you go. Fishing analogies again.
That's good.
Very cool.
The next one from David is about the same paper. The Bornavirus paper, really? And he said, I never heard of a paleovirologist before. He sent 2 different articles about this, one from Slashdot and one from Science News. And he said it's interesting to see the different takes from the same article. From past podcasts...
come on. They just determined why King Tut died. Come on. He died of malaria. He died from malaria.
Problem was that his parents were brother and sister, right?
No, he died of malaria. They found the genome of malaria in his own.
How do we know he died of it?
Paleobiology. That's paleobiology, because he was 16 years old, for Christ's sake.
Anyway, paleovirology is the same thing, right? Looking for viral fossils. Exactly. And where can we find them except in genomes? It's the only place we know.
Correct. This is exactly right.
He continues, from past podcasts, I've gotten a fair idea of how viruses differ from simpler entities like prions, plasmids, and viroids. I don't have as clear a picture of how viruses differ from some of the simpler bacteria. Would that be rickettsia? Other than the protein coat, are the more complicated viruses, mimivirus, that differ in complexity than whatever, are the simplest bacteria? Are there life or life-like biological entities that are somewhere between viruses and the simplest bacteria? Wow.
Yeah.
You're taking us to the edge of our knowledge.
Who wants to tackle that?
Well, one of the first things that comes to my mind as a sort of a distinguishing a distinction between viruses and the simplest of bacteria is ribosomes, right? I don't know any.
Yeah, right.
So Rickettsia have ribosomes, don't they?
They do.
I hope.
Rickettsia have everything they need, apparently, except, I mean, they have everything, but they can't live on their own, right?
No, they can't.
But they have energy generating apparatus. They have, membrane generate. They have enzymes that make lipids. They have ribosomes, a protein synthetic machinery.
Right. So viruses are missing a lot of that stuff. They can't metabolize lipids. They can't do their own protein synthesis. They can't do their own energy metabolism. Even a guy like Mimi virus may have some proteins that contribute to those some of those intracellular processes, but they can't do them themselves. And then I think you got a big jump to something like Rickettsia, where all those things can happen. I don't know of anything in between.
No, I don't either.
No, I don't either.
Mimi is thought to be right in between, but it's very far away from a Rickettsia even.
Right. It's still very virus-like.
Yeah, I mean, it's got a huge genome, but it still doesn't have a lot of stuff. So
can't do its own energy, can't do its own protein synthesis.
But maybe there's something out there that we haven't found.
Could be. Keep
looking. Okay, I think we'll stop there because the next one we could talk about for an hour.
Yeah, that's a whole show. That's a great. I didn't know anything about that.
Fabulous.
That is really cool.
Oh, I can hardly wait then. Email it to me, please.
So we'll do it. We'll do it next week because.
No, I must tell your listeners though that I'm standing at my window looking out over the wilds of Fort Lee, New Jersey. And the sun has just come out, and it is the most beautiful setting of a wintry scene I have seen in many years. It actually looks like a Currier and Ives print of a little village.
Take a picture of it, and we'll send it to us.
I'm going to do that. I will do that, and I'll put it on the internet, and I'll send it over to you.
Send it to me. We'll put it on the podcast, just like we put a picture of you fishing last week up,
Oh, my God.
It's the one you'd sent me where you had fallen face first into the river.
Oh, that one.
Yeah.
Where the fish was so big it dragged me down the river.
Exactly. All right, let's get on. Let's do some picks.
Oh, okay.
Dick, you had a movie you were going to pick.
Yeah, well, there are two of them. One is called The Devils and the Doctors.
You already picked that one. You picked that one already. Oh, that was for TWiV.
In the past? Well, I already told you about 2010, A Space Odyssey, but my pick of the week is actually from Science Magazine this week. On the cover is this remarkable picture of a construction. Art and science tend to form a similar bond when they're trying to demonstrate the intricacies and complexities of life using inanimate objects like plastic, those plastic binders that you can use to hold things together with. It's got a zip lock almost on it, and you shove one end through it, and then you can pull it tight and hold something together. Well, they've designed the inner workings of the lung. The cover picture on this week's Science shows that, and inside these are the contest winners for illustration. And it's a remarkable set of I don't know what to call them. They're not just illustrations; they're demonstrations of scientific principles visually. All right, just by looking at one thing, you get a whole concept.
We actually have the same pick of the week this week.
You're kidding.
If you're picking, if you're picking the winners of the visual presentation contest, is that what you're getting at?
2009 Visualization Challenge winners. That's correct, and that's my pick.
That is exactly my pick of the week too.
It's okay. So, you can both have it because it must be really good then. It
is pretty cool.
Wonderful.
So I've now copied and pasted.
They're not typical. They are not typical pictures.
No, they're this one. Unlike the previous, we've done these sorts of things before, where we picked the Nikon and Olympus both had imaging contests. This one's a little bit different. It's the National Science Foundation and the AAAS do this annual Science and Engineering Visualization Challenge. And there are some photographs. That's one category. Then they also have a video category, a multimedia category, an illustration category, and some others. And they're really, really. It's a neat assortment of stuff that's all centered around. They're all gorgeous, and they're all centered around the idea of presenting information in interesting ways.
And this is so cool. I love this stuff. Look at this.
Yeah, I do too.
Wait a minute. What's this hamburger? Oh, it's
The jellyfish hamburger.
Yes. Yeah, there's an autoplay. There's an autoplay video on the homepage.
Just keeping your hands off the wildlife.
Yes.
And the one about following a dollar bill throughout the United States is just precious.
Yeah,
I mean,
the graph from the Where's George site. Yeah.
Wow.
you know, at the end it said, it says, oh, where the hell are my glasses here? Yeah, I'll get this straight. It says, one of the judges said, I like the extreme geekiness of it and how it made me want to watch it again to absorb it all, says Judge Thomas Lucas. It was so rich. Just finding out what happens to your money. This is a way of tracing a single dollar bill throughout the economic system of the United States. It's quite...
And it has a larger
precious.
point. It has a larger point, too, because it traces human mobility. Because somebody had to go there to move the dollar bill. So you're
sure.
a graph of human mobility in an industrialized country, which is useful for a lot of things.
Said that we're going to adapt this for viral outbreaks and stuff like that, too.
Absolutely.
So we're going to start watching this. It says Grady hopes the video will stimulate other people to think of new ideas as to what to do with these data. They've already started using it to study how diseases such as H1N1 spread, and linguistic professors want to compare these travel boundaries to dialect boundaries. So, wow. Hot stuff.
Good.
And it attracts your attention and you can't stop looking.
A double pick. I actually saw this and a copy of Science lying on a desk the other day, and I said, that would be a good pick. But I didn't pick it. It would have been a triple pick.
You know, Vince, your nose just got longer. You know, I hate to tell you this.
A trifecta. No. My nose never gets long. No. Rich, what have you got?
Okay, so I'm going to stretch the boundaries of Science Picks of the Week again and go for science fiction. I'm recommending the novel Foundation by Isaac Asimov.
Oh,
now that's just a start because Foundation is the first book of the Foundation trilogy. And then, in fact, that was written in the 50s. And then after the trilogy was written, there were prequels and sequels. And in fact, there's stuff that ties all of his novels together. And you can, I've supplied a link here to a Wikipedia thing on the Foundation trilogy as well. You could start by reading the prequels and stuff, but I think the better way to do it is chronologically and read the first book that was written, which was Foundation, and see what you like. And I'm not even going to say anything about it because I don't want to spoil any of it. Okay. And in fact, I wouldn't even read the wiki site.
No, that's right. That's right.
Go read Foundation, and then if you like it, go to the wiki site and find out more about what this is about and what order you want to read it in. But Asimov was terrific, and this is a wonderful series. I read these during, if I recall correctly, during my first year of graduate school instead of studying for exams.
Well, you remember he was a biochemist too, so I mean, he had a lot of a big science background. So
I think he actually wrote some science nonfiction as well.
Yeah, lots of it. He wrote hundreds of books.
Yeah,
he
so
was
there you
amazing.
go.
Cool. I read that years ago. Boy, I might have still my old copy of it.
Yeah.
This is great. Well, my pick is also a book. My pick is Natural Obsessions by Nathalie Angier. And I don't know if any of you remember this, but I
read it a long time ago.
Back in the 80s, she went to the Weinberg lab at MIT, spent a year there, and she wrote about what she saw. And it's not all pretty, but it's life in a lab. And if you want to see what it's like, if you're thinking about doing science, this is the book to read because it's all bare. She doesn't hide anything. Read this a long time ago. I knew Bob Weinberg. He was one of the first guys I met when I went to MIT. He was a very nice guy and does good science. And I have to thank Rachel in my virology class for coming up after class one day and suggesting this as a pick of the week. So thanks, Rachel. Nice. It's a great pick, and people will love it. Nathalie Angier, of course. She writes for the Times also, right?
Yeah. She's brilliant. Yeah, she writes in Science Times and also Science Magazine.
Good writer and a great book, so check it out. And that will do it for another TWiV done remotely all around. Dick's the first remote TWiV you've done.
It is.
A little muddled, but
it may be the first cell phone TWiV.
It is the
That's
first
right.
cell phone
First
TWiV.
cell phone TWiV.
Yeah.
Well, we'll get better. We'll get better. Don't worry.
I will teach you how to do Skype, but I'll come over and set you up. But you know what the cost is, right?
I'm afraid I do. I do. It's a home cooked meal, and you've got to be kidding. I mean, we'd love you to come over.
Dick is a great cook, guys. He's amazing. I was at his house for dinner one night. He is incredible.
Is there going to be a TWiV party sometime with Dick cooking?
I think so. I think we should do this.
This is a great idea. I think it would
we
be
should
fun.
have TWiV recipes because I like to cook.
Yes, we can do. You know what? Your recipe of the week, your pick of the week. We can do all of this.
Well, Dick has written a cookbook too.
In fact, you know what I've got? No, wait. I can offer this as a prize to our listeners. I have a cookbook. I've written a cookbook. I will share that with the guys, and you can tell me what you think of it. It's called Chef as Scientist. I believe that's what I called it. The Chef as Scientist, something like that.
All right, pass it on. I'll try it.
You'll see. It needs improving, but it's okay the way it is now.
Well, I think we should have a TWiV dinner. We should all attend, and we'll record the whole thing and put it up.
All of our listeners can join us.
That's right. We'll stream it live.
We'll be held at the Waldorf Astoria, December 30th. Be there.
All right. Gentlemen, thank you. Thanks, Dick, for checking in.
Okay.
Good to hear you. Medicalecology.org is where you can find. And Trichinella.org. And check out TWiP, microbeworld.org/TWiP. Alan Dove is at dovedocs.com. Thanks, Alan.
Always a pleasure.
And Rich Condit, you can find down in Gainesville, University of Florida Microbiology Department, or Molecular Genetics and Microbiology, right?
That's right.
We changed
home of the Fighting Gators.
They have a good team, I think, right?
They do. We nearly did it. Nearly did it again this year, but now we got to rebuild.
We watch.
I think the science is better. Rich, isn't the science better than the football down there?
The science is better than the football. The science is terrific.
Oh my goodness.
All right. And does anyone else need to be thanked? That'll do it.
Thank all of the people who wrote in their emails.
Yes.
Oh, absolutely. We love them. We love them.
We didn't even get through them all. It's just wonderful, wonderful questions.
Yeah. No, we have more, and we try and catch up periodically. If you like this, tell us. If you don't like it, you can tell us too. But you guys ask great questions. We can do whole episodes on your questions. And keep sending them to TWiV at twiv.tv. If you've listened to TWiV for the first time, go on over to iTunes or the Zune Marketplace and subscribe and leave a comment. That always helps us. You can also find us at sciencepodcasters.org, promednetwork.com, and microbeworld.org. What a media empire.
I tell you.
Thanks for joining us. You've been listening to This Week in Virology, the podcast about viruses. We'll be back next week. Another TWiV is viral.